let's review the genetic code.
Every gene in a chromosome has information to produce a protein.
And DNA is like a recipe for those proteins.
Right. The sequence of the nitrogen bases in a DNA molecule determines the sequence of amino acids in a protein. Remember, RNA polymerase enzymes read the appropriate section of DNA and then build a messenger RNA strand as a result. And that brings us to the triplet code. Do either of you remember what a codon is?
A codon is the messenger RNA sequence for 3 nitrogen bases that code for specific amino acids.
And with 4 different nitrogen bases, there are 64 possible combinations of 3 nitrogen bases, which make up codons, and code for specific amino acids.
Right, because 4 to the power of 3, or 4 cubed, is 64. But there are only 20 amino acids, so that means some codons code for the same amino acid.
And they can also signal the beginning or the end of this translation.
Right. Let's build a short 9 amino acid protein. And we'll use this DNA here. Actually, Peter, can you put these last 2 on?
Yeah.
Thanks. As our code. What would be the first step?
Well, the first thing that we'd have to do would be transcription. And for that, we need another mRNA strand, which would match up the codes on the DNA strand. Like this. And on the end, we would have a terminator codon.
From this mRNA strand, specific transfer RNAs bring specific amino acids to the ribosome, and a specific amino acid chain is built.
So why don't you line up codons with the amino acid?
Okay, they would all match up like this.
Okay, good. And as you said before, AUG is an initiator codon, and UAA is the terminator codon. Let's see what happens when we replace one of the bases in the DNA molecule. It's called a base substitution mutation. Okay, take a look at the codon for arginine, and look at the corresponding DNA segment.
GCA?
Right. Now replace this C with that A.
Okay.
Okay, what happens to the codon now?
Well, it should have another U.
There you go. Okay, if that change were made in the DNA, would it be the same protein?
No, because the codon would now code for leucine instead of arginine.
Right. What we've put out here is a sequence of amino acids. The sequence of amino acids is important to proteins. And we've put out in a line here that each of these amino acids is a 3-dimensional structure. The sequence of those 3-dimensional structures determines the overall shape of a protein. The shape that a protein assumes determines its function. Now, there's another 9 amino acid protein that we can look at. The first one that we made here called ADH. And what I'm putting out here now, actually, can you use the leucine from that other mutation?
You bet.
Right. Okay, this is a molecule of ADH. It's 9 amino acids long. This is a molecule of oxytocin, also 9 molecule long. And the only difference between these 2 molecules, we saw in the leucine, the 1 base substitution, and isoleucine instead of phenylalanine. Now, both these molecules have very different functions. They have very different shapes.
Okay, so if we change the sequence of the amino acids, then a different protein is produced, which has a different shape, which would mean that it also has a different function than the original protein without the mutation.
Absolutely. Another kind of mutation, and it involves the addition or deletion of 1 or more nitrogen bases in the DNA sequence. Let's take this blank piece of paper and let's delete this G from the DNA. How would that affect our protein?
Well, it would cause a shift in the way that the DNA is read. That means that the C would go with the 2 A's, and because it needs to be a triplet, and all the other letters would change too.
Right. The ribosomes simply read the information in groups of 3 nitrogen bases. So the addition or deletion of a base means that there's a shift in the frame of the 3 bases. That's why it's called a frame shift mutation. Now, this can also happen when a piece of DNA becomes separated from the chain. X-rays can make that happen. If repair enzymes don't splice that DNA back into the appropriate spot, then there are dramatic changes to the genetic code.
Yeah, I can see how frame shift mutations could have such a major effect. I mean, they create a completely different protein. But base substitution doesn't seem to be all that dramatic. It just causes a single amino acid change.
It could still have lethal effects. Have you ever heard of sickle cell anemia?
Oh, that's when you have a normal red blood cell, and then some of the cells are shaped like sickles.
Right. That change in shape is caused by a single base substitution mutation. A thymine is replaced by an adenine in the triplet that codes for the 6th amino acid of the beta chain of the hemoglobin molecule. That single base substitution completely distorts the shape of the red blood cell and makes it much more prone to clotting. There are lots of other serious side effects as well.
So the original DNA triplet would be CTC, and the mutated DNA would be CAC.
Okay, so the triplet CTC coded for the mRNA GAG, which in turn codes for the amino acid glutamate. The mutated triplet CAC, which codes for the mRNA GUG, which creates valine.
Right. Sickle cell anemia can be lethal if the person is homozygous for the sickling allele. The replacement of glutamate by valine causes red blood cells to assume an abnormal shape. This sickling of red blood cells in homozygous individuals leads to obstruction of blood flow in capillary beds and small vessels. Poor oxygen and nutrient transport and consequently poor tissue development are the result. In turn, these factors may result in bone, lung, or intestinal damage, kidney failure, brain damage, and even heart attacks. These are pleiotropic effects, when one gene affects many characteristics. Damage to that gene results in many symptoms.
There is really a heterozygote advantage, and that situation we know has occurred in things such as sickle cell anemia, where if you go to Africa you find a high ratio of individuals with sickle cell anemia, and carriers of sickle cell anemia in fact have some protection from malaria. So what you need is 2 things. One, you need some individuals in a population that carrying, and you need some environmental selective criteria that make those individuals viable and those that don't carry the gene.